Let's get started. Our speaker now is Johan van der Koppel, and he's going to tell us about phase separation as a lens to view patterns in ecosystems. I will be appreciated, Brett.
Thank you. Thank you for the organizers to invite me. That's been, and of course, thank you all for still being here. The last speaker of the conference and listening to a rogue ecologist among all the mathematicians. It has been a really interesting week for me. I must admit, I only understood half of it. But there was really interesting stuff in some of the talks, which actually applies to all my own work. I will actually point to that. And so the talk is by me and by my former PhD student, Quan-Xing Liu. He's now in China, as you can see. Combines much of his work and thinking, so I put him also as a co-author on the list. So, talking about pattern formation. Well, wonderful things happen if you don't have humans in an ecosystem. Then you get all sorts of nice things happening, like arid vegetation patterns, as you see there in bushlands or coral reefs in the Great Barrier Reef. You see like these bands, these Turing-like stripes, but mention also this on two scales, like small scale and big scale stripes in this landscape. Of course, Safting and salt marsh in the Netherlands is also a very pattern system. We actually recently published a paper where we basically argue that this is a nested Turing pattern. So simple principles can come back even in very complicated landscapes. Or mussel beds, my sort of pet landscape, also makes nice patterns. I won't be much talking about that today. But first I want to give you a little background about pattern formation in ecology, in ecosystems, and the theory behind that. And it started mostly with what you see here. There's an arid vegetation south of the Sahara. You see, bushland. So these little balls that you see are bushes, like 5 meters across, 5 meters high. Gives you a bit of sense of scale. And in this vegetation, all these holes start to appear, as you see here. Basically, in these holes, it's basically desert soil. I mean, it's really hard, rock hard. It like feels like concrete. And but under the bushlands, it's completely different. And to make a sort of an understanding how this works, and of course, it's a dry land. So water is a limiting resource. It determines to a large extent how this ecosystem functions. So rainfall comes down, it lands, and particularly if it lands in the spaces between the bushes, the infiltration is really blocked by this compacted soil. The water starts to move then. If it rains in Africa, it really rains. So lots of water starts to go overland and ends up in these bushes. And the roots, but also the whole ecosystem in the soil, opens up the soil and makes the water infiltrate. So the plants facilitate themselves and each other by promoting the infiltration into the soil. Well, and of course, the water moves, the overland flow moves. So you can already sort of imagine a sort of competition for the water between the bushes. So this competition on the one hand, on large scale, small scale facilitation along the trees gives you a configuration for pattern formation. As you can see here is an example of a model. There's many models that sort of depict that. And a classic form. And it basically follows, the people making that, we're not the first to make a model of a pattern. It follows the model made by Turing for the, development of patterns in the sheet of cells. That turned out to be much more complicated, as you all know, but it works very well for ecosystems. So you have a sort of short-range facilitation, long-range inhibition by the depletion of the water, and that leads to a situation where a small little perturbance with a certain wavelength gets amplified because there's more growth in there, and creates a pattern. And of course, this is very much a growth, that's an important thing, it's very much a growth-driven pattern formation. You have more growth here, less growth there. And the substance causing that is rainfall, and basically rainfall goes through the system, it's a flux. And so, I want to take you now to a different ecosystem, a different example, where things turned out to be just a little bit more complicated. And this is a mussel bed. It's, in the Netherlands, my pet ecosystem. And it makes, interesting is it makes all sorts of patterns. If you look at a large scale, this is about 200 meters wide up here. And so this, it makes sort of zebra path patterns, as you can see on this photograph of a mussel bed. But if you look within these patterns, then you see that it's not a scale of patterning. So what is it about patterning and mussel beds? Why do they make patterns? Well, the reason for that is that it's a dangerous world. I mean, they're basically growing on a tidal flat. They're loose. There's nothing to attach to. They're on the sand, and there's birds flying around, and they want to eat you. And there is wave action crushes you from the tidal flat and throws you into the deep, where life is more dangerous. So what can they do about that? Well, they stick to each other. They glue to each other by actually viscous thread, a sort of a glue, and make networks, make whole mats of muscles. And that's, of course, an effect from one muscle to the other. It's basically just a direct facilitation from one to the other. But there's also something else. There's also competition. And of course, they're filter feeders. They eat the algae from the water, consume that, and the water flows. So you can imagine that of course, they all compete for the same amount of algae in the water. And it's a long distance effect. So on the one hand, facilitation is a short distance effect, and competition is a long distance effect. And we call that scale-dependent feedback. It's just a name for Turing's principle applied to ecosystems. And yeah, you can make models of that, and models looks very similar to what you also can make for arid systems. And the model is actually quite similar. And if the water flows up, then this interaction creates this banded structure. As you can see here, I don't want to go into too much detail because this is a bit of an introduction for me still. And the patterns that you then observe look pretty similar to what you see on the photographs. And so, yeah, this is the big scale patterns. Well, the next step is, of course, these small scale patterns. And that was actually an interesting question. And when we were looking and making these photographs, we were shipped around by a fisherman. He had a boat, and he could bring us there. And he says, like, yeah, you scientists don't have to study this. Us fishermen, we know this. We know no need for scientists. Just take mussels, you put them in a bucket, make them loose, put them in a bucket. They make these things right in front of you, these strands. I found that really interesting. I thought, if that's true, then you basically have a sort of an experimental system for self-organization on ecosystem scale. That's pretty neat. So yeah, I went home, got myself some mussels and put them in a big container, put a webcam over it, and got nice water from the nearby bay, so to say. It's sort of a sea arm. And spread them out as homogeneous as I could. I had this idea of Turing, symmetry breaking instability, homogeneous as much as you can, and then see what happens. That's the idea. So here they are, and then here they go. This is, of course, an accelerated movie. It speeds up. It's about 5 hours of time that you see, and they make patterns, as you can see. And you can see that they also keep moving. They keep on looking for a better place within this little bunch of mussels. And you can also change the density, and then it becomes more. The wavelength doesn't really change much, but the little, they get more detached from each other, and the clumps get smaller. So it looks nice. It looks very similar to what we typically see in many models. And people always ask me, if they see these movies, it's like, well, how do they move? If I have them on my plate, they don't move at all. So how do they move? So I included this movie in here. So you see here a mussel moving, going with his arm. And so it has a foot or arm or whatever you call it, tentacle. And it actually does all sorts of things. It moves, and then it washes, it feels itself, and then starts to feel around, and it moves a little bit again. And this sort of behavior, sort of composite behavior, turned out to approximate a levee walk. And we found it very interesting. So this is a little detour from the rest of the talk. But basically we found out that, first of all, they are quicker into getting into the patterns with this levee walk, but also the patterns form quicker if they do a levee walk. So basically it's a feedback where they are adapted to the landscape that they themselves make. This was basically what Monique de Jager has been working on.
Are they on a solid substrate with water around them?
Yeah, this is all on a piece of rock or a piece of sheet or whatever. This is plastic, I think. In my former movie we were on a street tile. And there's water coming straight from the sea, being pumped over them slowly. So it's always fresh water. It's very important because otherwise they just well, it's complicated. Yeah, that were the conditions.
Change if you change the rate at which the water is flowing?
The water flows really very slow. So it flows, but it's really, well, you can see things moving around. I think the pumping of the water by the muscles creates more movement in the water than the water being pumped in. So you want to have a very slow refreshing. Okay, go back to so this pattern formation. So we thought like, well, we need to study this. It's a very nice experimental way of pattern formation. So can we sort of tease out how this works with this by studying the behavior? So basically we started to follow the muscles, as you can see here, and looked at how they moved as a function of the density of other muscles around them. And well, we did a lot of study on that. I also want to keep this short. It's basically an extensive introduction. And this picture actually gives the nicest, let's say clearest depiction of what we actually found. Is that we, at some point we thought like, well, it's all messed up. We need to separate the densities, a clear result on how the movement responds to density. So we made different densities, like two of them, almost alone. This is 8, this is 32. This will be almost, maybe a little bigger than a normal batch. Well, this is 128 muscles. There's always a power of 2, you can see. And this is way too big. There's too much muscles. And then we looked at how they moved, and as you can see, it forms a sort of a parabola shape. So they move a lot when they're alone. They move less when they're at 8 or 32, and they move more again when they're at 122. So you get the sort of a parabola shape. Of course, they don't move out of the patterns, but they just move, and then the patterns just start to disintegrate and become basically elongated and smaller.
Is this the movement of the cluster as a whole, or like of the single?
No, it's a muscle in the cluster. And always 2 muscles, because otherwise you get sort of bias in statistical bias. And so always 2 muscles to study per cluster. Okay, and so, well, we took this into the, to cut the paper short, that made a very nice study. One of the, I think, clearest example at that stage of pattern formation in an experimental way in an ecosystem. So we got it into a nice journal. I'm very proud of it. It's my only first authorship in science. So it first put out, but then after a little while it started to itch because I was getting doubts about this paper. And so because we said like, it's like Turing's process is doing this. And at some point I said like, well, I don't know, and they move. There's no movement in Turing's model, so maybe it's not that accurate. And that was one thing. And the second thing was basically that nobody cited it. It hurts a little, you know. And so you think like, what's the cause of that? And I thought like, that's because it's an individual-based model. They're all unique. It's difficult to compare this. So we need to make a PDE model of this. You had a question?
Yes. Do they also move by pulling on each other or only on the substrate?
All of that. And now, if you're in the sand, the small ones can move on the sand on their own. If they're bigger, that becomes harder and harder, so they pull on each other. But I've also seen that one muscle made a wire to a second muscle. The second muscle probably didn't like that, started to move and pulled the other one along with it. So I didn't measure that one. I thought I would have pseudo replicate. And so basically we started to think about, can we make a differential equation version of this individual-based model? Well, for you this is child's play, of course, but well, we start with basically a differential equation model. The classic one is Fisher's model in 1937 with diffusion in it, but of course diffusion only spreads it out. So yeah, we thought like, wait, we need something different. We need something that has actually a diffusion that is dependent on the density, as you can see here. Well, this was picked up by my second author, Quan-Xing, and he said like, well, let's see whether we can make that this translation. Can we translate the individual movements to a population model? And of course you don't need an F because there's a fixed amount of muscles and it only happens within 1 day. So there's no reaction term, it's just basically this. The D, of course, is a bit more complex because it's a population flux, not an individual movement. Luckily, there's lots of theory, and I always point to Einstein here. You guys say like, oh, and lots of other people also doing that, but I want to get the ecologists to read Einstein, so that's the reason why I read Einstein. And it allows you to translate the movement of the individuals to a population flux. And of course, you can see the population flux can become negative. So it becomes aggregative rather than diffusive. And at first, Quan-Xing was trying to do this, and it didn't want to work. Didn't want to work because they're aggregating and aggregating and aggregating, making basically mathematical piles of muscles, and then still aggregating until it was like a muscle black hole. And so he said, like, well, but at some point he had the solution. He said, like, I need to add this. And you guys are of course saying, oh, of course. But for me, this is like the derivative of the derivative of the derivative of the derivative. I mean, I said, okay, whatever you do, put it in. And this was the model he came up with. And yeah, it's behaved like this, like a phase separation model, of course.
Muscle.
Sorry?
Muscle limbs.
Muscle. Yes. Well, patterns. And I thought, well, where did you get this for the term? And he said, like, well, Wikipedia is just in the Cantor equation. So about phase separation, I had heard of it, but I didn't know much about it. So I learned then from that it's basically it happens in alloys. You have iron, and you have iron and carbon. You heat it up, it becomes iron carbide, and they don't like each other very much, and that creates patterns. And of course, that is also it's a bit of. I want to try out an explanation that I give to ecologists about how this works with free energy. So that's maybe something nice to consider. So basically, I mean, how do you explain energy wells, the double well potential to an ecologist? Basically, imagine you have Republicans and you have Democrats, and you mix them. And there's full of energy then, because the sparks are flying and there's lots of work to be done if you don't watch out. They storm the Capitol. And so, but then you have bubble formation, and they all live in their own bubble, and they don't see each other anymore. You have phase separation, Republicans and Democrats, and they're all living in their own little bubble. Tension is much lower energy levels. We can have a discussion about this later, whether this actually makes sense, but it does do the job with the ecologists. So, yeah, what next? Oh yeah, sorry, I'm going too quick. So we thought, cool, we can, actually I must say, we were really excited, because we thought like, we have a biological version of phase separation, isn't that cool? And then we looked in the literature and we found Michael Gates. Okay, not as new as we thought, but still, it's much bigger than his work, a much bigger scale, so we still thought, cool. Okay. Yeah, and then we submitted this, and it was, well, lots of comments, but there was one comment which was made by a reviewer. We didn't know who it was at that stage. He said like, yeah, you can say this, phase separation, but Quan-Hilliard predicts that you follow the Lipschitz, the coarsening process, the formation of these patterns, follows the Lipschitz-Liozov law. I've never even heard about that. So, and my first response was like, yeah, but this is animals. This is real world, not mathematics. You don't get this. So we did all the other things, not that thing, and we resubmitted it, and we got it back, and I think we were really lucky. And basically saying like, if you don't do this, we're really gonna reject it. So and then we thought like, oh, okay, there we go. We have to do it. And so at first we just looked at the model, and the model followed it pretty okay. Oh yeah, I should explain, of course, Lipschitz-Liozov law means that the wavelength increases power of time, as you can see here with the function of w to this t to the 1/3. And so this is what we tried, and the model did this very nicely. And the muscles, as you can see also. And now, of course, we didn't know the theory, so we nicely put our hands over the part where it got flat. Because before I came here, I had never heard about arrested coarsening. So I learned something to you guys here. So they stop at some point. And we think, of course, that's because of, in ecological sense, it would be because of food limitation. If the patches get too big, the ones in the middle don't get any food anymore. And we actually tested for that. We showed that if we have a patch and you give it no food, it splits up. If you give it food, it stays where it is. But of course, it's not that we stop giving them food at a time period of about 100 minutes. So maybe it's also built into the muscle that when they have a certain size that they stop moving. I don't know. We are looking at movies, so this is what we know about it. Okay, so yeah, and then also a nice paper. I must stress the work of, actually, Quan-Xing, you and me, we didn't get the model right. It was not consistent. It was actually Arjan Doelman and Vivi that got the model to be precisely correct, and they also did most of the mathematical analysis that's in the appendix of the paper. So I should stress also their contribution. Okay, so yeah, nice paper. Of course, I first sent it to Nature and Science and so on, but I think that they thought like, you don't fool us again. So, but now it's also okay. But to summarize, so basically we come up with a different approach to pattern formation, because at that stage most of the papers were about Turing patterns, and actually I think there's still examples of pattern formation in ecosystems which were brought in forward as being a Turing pattern, which I think, I'm not so sure about that. And so, well, the difference of course is that Turing's model is growth driven, and basically there's no conservation of mass of any sorts, while in what we call movement driven self-organization, there's just aggregation of a fixed amount of substance into these patterns. And so two different types of pattern formation for ecosystems. Yeah, so then the question of course is this only valid for mussels? No, not. We have two categories. One is like full of examples, and the other one is like empty with one example. Mussels, not a very strong theory. So other possibilities. Well, we think actually that the requirement for mass conservation is, well, let's put it like this: it's never perfectly there. I mean, you have ecosystems. Let me start again. For instance, phosphate is a typical substrate that's very important for growth of plants. And there are ecosystems that have patterns, peatlands to be precise, these ones, which are thought to be, peatlands, which are thought to be driven with phosphate. Well, of course, there's a little bit of phosphate coming from the subsoil, and there's some of it lost through drainage, of course, but for the most, it's just a stock of phosphate in this system. So it's almost mass conservation, you could argue. And so, yeah, many ecosystems might actually be very close to mass conserved. So we thought of, can we make a model which is then, just take one step back. So we already mentioned the arid system are basically flow-through systems. Water enters, it probably evaporates within a few days after raining down. So it's no mass conservation in this type of ecosystem. Let's take a few examples where it's different. And one of them, of course, is you can have dunes, or you can have, in this case, seagrass beds. Seagrass beds are really, they like muddy sediment. And what they do also is they protect, if there's mud in the water, if it drops down between the seagrass beds, the seagrass basically protect it. But also it gives them a better substrate, it gives them nutrients. Mud is important for seagrass, and you see that if you would walk around here, that the patches of seagrass are much more muddy than the parts in between. That also explains why the parts that don't have seagrass are sort of lower, because of about 10 centimeters of mud building up in the system. So here you basically have, in the whole of this area, a certain amount of suspended sediment in the water, while which is exchanged with deposited sediment, and that's basically triggered by the seagrasses. Another example is what I already explained, the peatlands, where phosphates is very important. They are taken up by the plants. They aggregate them into this, because there's amplified evaporation of water in the bands with the trees. These are trees, I should explain. These are little sort of peatland pools, you could say. That's mostly peat. And what you see here in between are trees. They're not that big, maybe some between 5 or 10 meters. Often pretty small trees. And the trees evaporate water faster than the sphagnum. And that makes the water to concentrate underneath the trees, accumulate the phosphates underneath the trees, and there's a sort of a cycle of phosphate happening. The leaves, basically, of course, and they drop the phosphates below in these stripes, making these stripes concentrations of phosphate. And so this is another example of such a process. And this one I want to do a little different. I want to give you a description of how this works, and you guys may guess what is the conserved factor here. so to say. So you can see there's cows, and they're not very natural. So a farmer brought his cows here in this field. It's quite close to the dune in the back. But you can see there's sort of two-phase effect of this very short grass, short grazed by the cattle, because they like the short grass. They don't like the rough and old, the high-density patches in the back, because that's much more spiky, much more lignin in there, much more silicates. And so that's the bad food. And that's what you see in front of it, this lawn, is the good food. So you see the two phases. And there's the happy cows. So what is the conserved factor in this system? Any idea? Did I explain this right?
Can you give options?
Well, there's short grass, there's high grass, and there's grazing, and there's cows.
The grass purchased by the cows?
The cows.
Yeah.
The farmers, they brought a fixed amount of cattle in the field. At least fixed on this time scale, which is irrelevant. But basically, I mean, there's a certain amount of cattle that they agree to. This is a nature reserve. Otherwise, the farmer would just bring as much cattle as can be so that they short graze everything. But here they want to have a mixed vegetation. And it's basically the amount of cows determine how much short grass you get. If you would take a few cows off, the rough patches would expand until the cattle are exactly the right amount to short graze the vegetation here. So it's cattle that does it. So basically they're big muscles, you could say. They aggregate into patches, but it's not themselves. It's their grazing activity that aggregates. Because you'll see them lying around in the middle of the coarse patches, but they don't eat that. Well, another example, and you could, well, I've already explained it, I think. You can basically make a model of moving herbivores and grazing herbivores, and two different movement modes. And so, this is actually, I think, the most important thing for me to say here is oh yeah, sorry, go too fast. What's nice and striking is that the patches in the grassland have a tendency to be very round, sort of suggesting that there's sort of a tension at the edge, that everything's sticking out, is being grazed away. So just like in phase separation, they have these very roundish patches. So how to model this? Well, Quan-Xing, one of my postdocs for a while, made a model, you can see here, with defining U as fixed particles, V as mobile particles, and said like, well, basically they diffuse, and there's reaction terms, F and G. And if you have a situation where basically U and V are exchanged for each other, and there's mass conservation, that means that F and G are, I mix up all the symbols, are basically equal but negative to each other. If the net effect is increasing, then it goes up and then is removed from the other component. So basically F is the net sum of these two arrows on that side, and V is the net of the ones to the bottom. It can be a sort of C. Of course, if it's sand, C is 1, because it doesn't change. But if it's taken up, phosphate being taken up by the plants, and you take the plants as a pool, you have to include the fact that there's a conversion of that into plant biomass, and that's the C here. And so, this is basically the basic structure. Koen, who simplified this into this setup, as you can see here, U and V, and then basically a single F, and you can, by minimizing, making the model non-dimensional, you can make it just one function, the F. If you think this is very similar to what Erwin Frey, was it? That was also striking to me, so I thought, what has Koen been doing? And when he wrote this down, he cites Erwin a lot, so basically just comes from his paper. But what Koen basically suggests is that for these 3 example ecosystems, you can come up with one equation that captures the dynamics of all these ecosystems. And he took basic models for these ecosystems and derived from that all 3 can be simplified, of course with assumptions and with all sorts of simplifications to this one equation. And that gives you basically the dynamics that you can see here. Looks all very similar to the other ones. So with coarsening dynamics and so on. And so basically what you have here is that the process of moving from the mobile pool to the fixed pool is captured by u times v. Basically, it means that they're determined by the rate of collisions, assuming that the fixed particles, for instance, if it's sand, then there's a plant attached to it, and the plant actually captures the amount of v by fixing it. And then there's a release of the substance, which is purely a function of u in this case, and that of course levels off. You have a certain amount of alpha times u, which would be a sort of a linear rate, but that rate is depressed if u increases, because it sort of fixes the sediment as u increases more. Is that clear? I hope. Okay. So we studied this. Of course, there was coarsening, as you saw from a movie, and you can basically see that you have a lot of patches, and as the system develops along this area, you see that any patch that is lower than a threshold, which is the average size of the patch, drops out, and of course then when it drops out, also the average changes. So this threshold is always increasing until at some point there's only one patch left. And so the classic dynamics of what you find in a phase separating system. To put it into an ecological context, we're still on part of an ecological European Union Horizon 2020 proposal, so you have to take it into much more applied context. So what if you have a disturbance in it? You have an ecosystem, and humans have disturbed it. And so you do this. Does it make a difference? You have your Turing system on that side, you have a density-dependent aggregation, a phase-separating system on the other side. Does it make a difference? Well, if you then run it in the model, you see that the Turing system very quickly repairs itself. The phase-separating system, not so much, at least on the spot. We removed the vegetation, but not the sediment attached to it. This is a system with sediment. And so the sediment becomes mobile and is captured in the surrounding patches. That has quite some implications for the management of ecosystems. So if you have a system that basically is governed by Turing patterns, you can try to restore it on the spot, stimulate regrowth and so on. But don't try to do that with a phase-separating ecosystem, because the resources are lost to the surroundings, and if you put a little plant in here, well, unless you make it a very big plant, you will probably, a very big patch, you will probably not manage to get this over the threshold. And so this will disappear again. So you have to approach systems that are phase separating in a very different way than systems that are following Turing's principle. Of course, on a very large scale, the amount of the patches just stay the same size. So on a large scale, you keep the same cover, but not on the spot. And this is, I think, an important message to ecologists and ecological managers. Okay, well, of course, to explain that, there's a very clear difference coming out if you look at how the growth of these patches relates to their dynamics. You see that in systems with scale-dependent feedback, so Turing systems, that the relation between the size of a patch and its growth is negative, meaning that any disturbance that reduces its patch size will increase its growth and it will just recover. The opposite happens with systems that are phase separating, is that actually if you disturb a system, you will move possibly the patch below this threshold. It's very hard to recover because its growth rate becomes negative. Okay, and so yeah, this is the last picture to show. It was also a paper in PNAS in 2023. So this is our latest work on this. And I think I'm finished with that. As a summary, so there's much more than Turing patterns. That's actually what I want to say to you guys know, of course, but ecologists don't know that so much. So we need more theory about pattern formation for ecosystems and some degree of mass conservation. The some degree is actually very interesting because there is no ecosystem with perfect mass conservation. It's always an approximation. You can approach that like assuming that the total amount of a specific resource is increasing. That's basically increasing the cover of changing the cover of your system. Or you can make it a function of its properties, of course, and then it becomes really interesting. You have feedbacks involved in that. And so it involves a fundamental change in pattern dynamics, and particularly with regard to resilience, as my last slide showed. And I think that integration of these physical processes and concepts in biology, well, would be a fertile ground for further study, I think. And, well, I hope I have sort of made you interested in ecosystems a good amount. Okay, to end with, I'd like to show you a picture of the Wannsee. This is one of my future projects. So we have models of all these creeks, also simplifying Navier-Stokes or shallow water equation models that describe how all these creeks works. But then, of course, the dynamics of if you have sediment in that model is just a little input-output dynamics. So what if you presume that the amount of sediment here is fixed, meaning that not everywhere things can grow, but only seagrass beds can only cover part of the area. Mussel beds can only cover part of the area. How would that develop? And that's something that we will study in a future project in the Wannsee. And I would like to leave it at that.
Thank you, Johan. Questions?
Systems, when you have a distribution of patches, you have distributions that are usually scale-free. But here, apparently, you have a characteristic scale of your patch. So, do you have a way to select whether you are looking?
Thank you for asking this question. I didn't bribe him. So basically, the scale-free patches you don't have in this sort of. Turing patterns, of course, have a characteristic patch size. They can be elongated and all sorts of things, but there's a specific patch size. It's a different patch size that you have from phase separating systems. I think Koen emphasized the log normal distribution for the development of patches. Well, first of all, as you can see, we studied this along. We did a classic phase space analysis. You can see here with the spinodal region and the binodal region. Our question was actually, how can you distinguish the changes in the patch sizes when you have just the normal dynamics or from a system where actually things are changing? Because you have patches are changing, but which would worry anybody in management, but can you tell whether it's actually a serious change or not? And so basically, what Koen came up with, and I hope I explained it in the right way now, is that if you have patch sizes that the Lipschitz-Jozef law is, it's been a few years since I've worked on this. But you get basically a gradient from when you have a small tau, global density, where the distribution of patches, which is these bars, is more close to what's predicted by the Lipschitz-Jozef-Wagner law, while if you move into the direction of B, as you can see there, it starts to be more close to the log normal distribution. And this log normal distribution is basically follows the scale. So if you would just have this system developing, you would see a certain patch size distribution moving. If you just simply divide that by the average patch size, they fall exactly on top of each other. And so that's basically the normal dynamics. And what you can see here is that if you move along this phase space, then you can see deviations from that. I hope I explained it right.
There's another question.
May I make a non-physics announcement quickly?
Yes.
They have found a lost key and card room key, and it's available at the registration desk. So if someone has misplaced it, please collect it before they close at 4:30. Okay, please continue now.
Go ahead.
So you show now that with the conservation law, it is essentially coherent dynamics. But at the beginning, you showed that the causing is arrested. So what is now the hypothesis in the context of these models? So how to get it arrested?
We didn't work on that. We just thought that must have something to do with the food limitation. So we didn't work any further on it.
I mean,
I actually got a theory for that.
Allen has one, I think, Fry. I mean,
yeah.
if the conservation law is slightly broken, then he selects a scale.
Yeah, well, of course, it's a fixed amount of muscles. There's no breaking in the conservation law. So it's a change in behavior of the muscles that does that. In this case. Oh yeah, here we are. It happens within a very small time scale of about, I think it's about, the total movies are a day, but I think what I show is 5 hours or something. And so you can see after 100 minutes it stops, but the same amount of muscles. Yeah. It must be a change in behavior. But of course that's on the scale of an individual-based model. How you translate that into the PDE, that's an interesting question, I think. Yeah.
You did perturbation in the simulation. Did you perturb your muscle?
The model that I show here was an individual-based model, so that's just a random movement.
The very last part where you erased the square region by deleting.
You mean?
This.
Oh yeah.
Yeah, do that with muscles.
Muscles, he's asking.
Eat them.
Well, of course we've done that. They move in, but to some extent, because moving is dangerous. So once they have made these clusters, then the longer you wait, the less eager they are to move into these patches. They like to be in these clusters, and that's sort of a rather non-mathematical explanation. But you should also, well, they are there, but they don't know that they are mic'd up, and so birds overhead, water flow coming in maybe. So moving is dangerous. But I do, well, the patches are never the same. They keep on changing. I've also done very long simulations, and they keep on. I think on the tidal flats, you actually see mussels making little towers. Particularly when they just find out that they don't have enough food to eat, then yeah, you can wait and die, and that happens actually because it starts with millions of mussels, and in the end there's about 1,000. When they're big, they get bigger and bigger and bigger, but the biomass doesn't change anymore on the square meter. So lots of mussels starve. And you see them sometimes making towers, and they climb on top of each other of about this high. And that's interesting. So maybe the mussel black hole wasn't that inaccurate. So that's. Although, of course, they just fall down at some moment. So that maybe is an explanation for the fourth order term.
The question in the front, and then Srinath.
Would this pattern of muscle change if, for example, temperature of water changes or salinity changes suddenly?
Well, if you put them in fresh water, then
environmental parameters. Yes, probably yes. But how?
If you put them in fresh water, they're saltwater animals, they will not move.
Maybe not completely fresh, but lower salinity.
We haven't tried that, but I think they will move less, be less eager. Oh, I expect a threshold in their behavior. Because of course they do encounter fresh water.
Stable. Yeah, you can check it in the lab experiment.
Yeah. The easiest way to change that behavior is to, well, the first thing I did was put a predator in. I put a crab in a cage next to the mussels, and that really stimulates them. Then I thought, like, poor crab, so I gave the crab for something to eat, mussels. Then he moved even more. The mussels moved even more, and then I thought, like, well, but it's maybe my hypothesis: do they smell the crab? Or do they smell the fact that they're dead mussels? So I just, well, smashed a few mussels with a hammer, put them in the water, and there they went. So maybe I guess they can smell all sorts of things, but they even smell that there's other mussels being eaten. So, but don't forget, these are just my observations, and also this, I didn't really test for this. It was not so yeah, lots of things to do, I think. Yeah.
Srinath?
Yeah, if you go back to your muscle data again, sorry to obsess about it, you have a small but somehow surprisingly believable one-third law. And it would be nice to check if the mechanism behind it is really what happens in Lipschitz-Dorsov, namely currents proportional to Laplace pressure gradient, with efflux therefore proportional to that. So are there ways of measuring that you're really seeing small drops evaporate, big drops grow? I mean, can you interrogate your data?
Yeah, it would be interesting to look at.
I'm saying that it's intriguing that even though your data range is small, over that range is a very good one-third law. So it'd be nice to know.
It's too dense. And maybe the second one is better for that. But if you see them keep on moving, and sometimes they cross, these sorts of things. But this is maybe too dense for that. Maybe the next one is better. This is a small top. Yeah, finished. So
I guess you need a thermodynamic limit of muscles. but
when they're babies, you nearly do.
Yeah. Maybe.
But Lifshitz spheres
you see,
seems to be
he was one
consistent
that
with the
disappeared.
bulk
No, it
distribution.
becomes a little.
I'm saying there's many more.
So this is easier probably to.
Maybe the distribution, the evolution of the cluster size distribution is something we didn't look at.
Post the muscle data on the on the internet. We will all provide an answer to you
Oh yeah.
in terms of the mechanism.
Sure.
Anyhow.
Yeah. Oh, if somebody wants to have it, no problem. Yeah.
Go ahead.
Maybe connecting to this morning's talk, so if you would have a mixture of crabs and mussels, would then be a non-reciprocal prokaryotic wave?
I don't think the muscles will stop moving. I don't think that. They will stick even more. So yeah, it would be cool if we could sort of get a traveling wave because of the. But I mean, the crabs are so diffusive, so movable. They will, in the time that the muscle, you would see the zigzagging over the screen here if you do that. Although they actually don't like it very much because they are afraid of predators. So they stay at the rim of this field. So you have to also find a way to do that, make it dark, and then put something on their back so that they are observable. But then you can't see the muscles anymore. Yeah. Well, I think it's a nice idea, but I think that you need, if you have a predator-prey system, so non-reciprocal interactions, and one of the elements is much more diffusive than the other one, the models would also not give you that sort of chasing behavior.
It will become homogeneous.
Yeah, exactly. Yeah.
Maybe you can, hurt the legs of the crabs.
There are also predatory snails. They move at the same rate, of course. Well, they're oyster-eating snails. But you see sometimes, if you see a mussel and there's a small little hole in it, that's a snail that dug a hole and then throws his enzymes in and sucks out the mussel. That will make it a gruesome story. But with these things, you can sort of try that. Although, of course, they will eat it and you will not have regrowth. Yeah.
Any last questions for our speaker? No, you have the last word now.
Thank you.